autonomic neuroeffector junction. Now, the autonomic neuroeffector junction is the area where that postganglionic neuron innervates the tissue in question, okay? And this is specifically cardiac muscle, smooth muscle, glandular tissue, adipose, okay? So think about the second neuron here in the series of two neurons. This is the second neuron. And so the relationship here at the tissue level is very different than a traditional neuron to neuron synapse. Because the second tissue is not another neuron, this is a different anatomy than at the neuron to neuron synapse. So the differences here is that we have varicosities. So varicosities are basically these swellings along the ending of the second neuron, so the postganglionic neuron. And instead of an axon terminal, right, we talked about an axon terminal, which is that large bouton, that large swelling, in the or at the neuroeffector junction, we don't see that axon terminal. What we see is a varicosity. It's just a swelling along the distal part of that neuron, okay? The second difference here is that the gap or the width between the second neuron and the tissue is much greater. We talked about the distance of the synaptic cleft, by about 0.5 to 1 brain fart, okay. So that was a smaller distance than it is for the neuroeffector junction, okay. The neuroeffector junction is going to be larger or wider than the neuron to neuron synapse, okay. Two important differences, the presence of varicosities and the larger synaptic cleft, or the larger distance. Okay. Let's speak about what happens at the neuroeffector junction. So let's talk about the steps of how an action potential can move along this second neuron and can excite this effector tissue. Now, this looks very similar to the synaptic communication process that we talked about in the last unit. We had an action potential traveling down this neuron because of the presence of voltage-gated sodium and potassium channels. Now, when we get to the varicosity, much like we saw at the axon terminal, at the varicosity, there are no voltage-gated sodium channels. Instead, there are voltage-gated calcium channels, okay? And so the first part of this is that the action potential gets to the swelling and we open up voltage-gated calcium channels. Calcium rushes into the axon swelling, into the varicosity, and this triggers the release of neurotransmitter, just like we saw in our previous unit, okay? Now, this neurotransmitter leaves the cell via exocytosis, or leaves the varicosity via exocytosis, and it's going to cross the cleft here. It's going to cross to the post-ganglionic side. Now, bear in mind, this is not another neuron. This is an effector cell, because we're not innervating another neuron. We're innervating a tissue, either cardiac muscle, smooth muscle, glandular tissue, or adipose, okay? So the anatomy of this synapse looks very different from a neuron to neuron synapse, okay? Now, when we release this neurotransmitter, depending on what neuron this is, okay, remember that our postganglionic sympathetic neurons release norepinephrine, and our postganglionic parasympathetic neurons release acetylcholine. So depending on what neuron this is, that will determine what neurotransmitter is stored in that varicosity. If it's on the parasympathetic side, acetylcholine is stored and it's released, okay? If it's on the sympathetic side, norepinephrine is stored and it is released, okay? Now, when this neurotransmitter is released, it's going to cross the cleft and bind to one of the receptors we just talked about, either an adrenergic receptor or a muscarinic receptor. Right, so it's going to bind to this muscarinic receptor here, and that will elicit the pathway that we talked about, okay? Whereas on this side, if it binds to an adrenergic receptor, that can be different depending on if it's beta 1, beta 2, beta 3, alpha 1, or alpha 2, okay? And so just understanding that norepinephrine will be released, will bind to this receptor. The response will look different depending on where this is, okay? But up until this point, it's going to be the same pathway. Now, much like what we saw with our traditional neuron-to-neuron synapse, there are also ways to clear the cleft of excess neurotransmitter. So it can either be degraded by enzymes, or it can diffuse away from the cleft, okay? It can also be repackaged, repurposed using that uptake molecule, right? So much like what we saw in our traditional neuron-to-neuron synapse, we can also have that happening here. So for example, the enzyme that is breaking down excess acetylcholine is called acetylcholinesterase, okay? Remember that ASE in the suffix means that it's going to be cutting or cleaving a molecule. So acetylcholinesterase is going to chop or break up or cleave excess acetylcholine, and that can diffuse away from the junction. Or we can have reuptake molecules that are splitting choline and acetate. Acetate can diffuse away. Choline can be recycled to make more acetylcholine, okay? And that's how that reuptake process works. Now, again, we described this earlier in a little bit more. We described this earlier, and here we're just filling in the details, right? We're talking about the name of this enzyme and the actual uptake molecule.